Ladies and gentlemen, I think we are almost ready to start. So welcome to this ordinary meeting and the lecture. My name is Anna Dominczak. I am a Vice President, Life Sciences of the Royal Society of Edinburgh, and I have a pleasure today to stand in for our President, who is doing a very good work for the society in China, representing society in China. So I've been asked to stand in, which is a great privilege for me. Now, first of all, as it is a public lecture, let me just say a couple of things about the Royal Society for those of you who are not fellows. The Royal Society of Edinburgh is Scotland's national academy. It was founded in 1783, which is quite a while ago, by the Royal Charter for the advancement of learning and useful knowledge. Each year, the Royal Society of Edinburgh enhances its position by electing men and women of great accomplishment to its fellowship, and there are more than 1,500 fellows currently who volunteer to do a lot of work for the society. And today, in a minute, we'll have a privilege to admit 2 fellows who were not able to attend the big admission ceremony of this year that was a couple of weeks ago. Now, one more thing I need to share with you is that after all these hundreds of years, for the first time in October of this year, we're going to have first ever woman president of the Royal Society of Edinburgh. And I am smiling when I say this. And she is Professor Jocelyn Bell Burnell, a graduate of Glasgow, who knows Scotland well and who currently works in Oxford and who will be with us for the next few years as a president. And I think it's a great thing, and I'm sure ladies in the audience share my smile. Now, first we have a bit of FRSE business, and the important business is that of 2 of our new fellows signing the roll book here. And may I ask Professor Peter Simmons, Professor of Virology, University of Edinburgh, to come forward first. Professor Simmons, sign the book whilst I say, in a name and by the authority of the Royal Society of Edinburgh, I admit you to a fellow thereof. Okay. This one. Right, and the second fellow to sign the roll book is Professor David Salt, Professor and 6th Century Chair of Plant Science, University of Aberdeen. Please sign the book, and I'll say, in the name and by the authority of the Royal Society of Edinburgh, I admit you to a fellow thereof. For posterity. Congratulations. Okay, and the second part of business is that I need to announce and read to you these scrutineers for the forthcoming ballot of the election of the new FRSE Council and office bearers for the session 2014-2015, and they are Professor Ian Deary from University of Edinburgh and Patricia Hinton, former Chief Executive of SIPA. And the outcome of this ballot will be announced at the annual statutory meeting on 13th of October 2014. And now to the main business of today, which is presentation of 2 medals, followed by a lecture by one of our medalists. So we have 2 medals, and they will both be collected by Professor Gadd. One is for him, and another one is for Dr. Nicola Stanley Wall, who is unable to be present but works with you, and both will be collected by yourself. Right? 2 medals. Fantastic. And we will now move to introduction of the lecture. I am delighted to be able to introduce tonight's speaker, Professor Geoffrey Gadd, who has been awarded the James Black Prize 2012 for his outstanding contribution as an international leader in the field of metals, minerals, and microbes. Professor Gadd occupies a unique scientific niche within Scotland, and his outstanding reputation has been achieved through work carried out in Dundee for over 30 years. He has markedly influenced the direction of his research field and has significant recognition in disciplines outside microbiology. The relevance of his work to understanding key environmental processes and their application is of wide public appeal. And of course, that's why we have a public lecture today and all of you to participate. We will have a question and answer session at the end, so please think about difficult or interesting questions. We'll have 15 minutes at the end to put some questions to our lecturer. I give you Professor Geoffrey Gadd for the lecture, please.
Great. Thank you very much for the introduction, and thank you very much to the Royal Society of Edinburgh for the Sir James Black Award. I'm very honoured to receive it, and it's a good honour for all the people in my lab over the years in Dundee who've contributed to the work, and also recognition for the field of research that I'll tell you about now, because as you probably know, life sciences today, it's a lot of biomedical, cell and molecular, and so this kind of work occupies a rather small niche, so I'm very grateful for this award and recognition of the subject area. Now, I've got some kind of connection with James Black. You might have noticed from my address, I'm actually in the Sir James Black Centre in the College of Life Sciences, University of Dundee. I did meet Sir James at a meeting in Strathclyde once on Scottish biotechnology in the early 80s, I think it was, where he chaired, and I can promise that all the slides I've got today, not one is the same as I used in that talk then. So there's the Sir James Black Centre in the top left, and every day I can go past this portrait that was made in 2006 when this building was opened. And in fact, that's my office right by the sign. So you can see how close I am to James Black, and that's the view out of my window that I probably see every day. So rather an unusual link. Okay, so geomicrobiology. So here's one of the Gary Larson cartoons, early microbiologists. So you can see them trying to look at microbes, before the advent of microscopes and electron microscopes and confocal and this, that, and the other. But if you take a look at the person looking at the wall, he knows perfectly well you don't need a microscope, to see the effects of microbes. I mean, that's an early geomicrobiologist. He can be fully aware of biofilms on the rock surface, the different colors, mineral transformations, boring, tunneling, dissolution, you name it. And so this is what geomicrobiology is. The roles of microbes as geoactive agents, their roles in the past, the present, and possibly in the future. And the usual kinds of things that you could think of in this context are mineral formation or dissolution, and amongst those headings are bioweathering, bioleaching, soil formation, because all mineral soil is formed from really weathering of rocks and minerals. Element cycling, all the big elements, the ones that we have, everybody has, in the majority amounts, metals, metalloids, radionuclides, but really all others, all elements of some kind of biotic component to their cycling in the environment. All these processes, metal transformations, are integral to all of them. On the applied side, both good and bad, some of the transformations we can put to use in bioremediation of metals, metalloids, radionuclides, organics, use of naturally occurring organisms to clean up pollution, to remediate polluted habitats. Bioweathering and biocorrosion might be bad biocorrosion, usually in the context of metals, bugs and things rotting, breaking things down, spoilage, you'll see. Bioleaching and biorecovery, to reclaim metals or valuable elements, something like this, and also for the production of biomaterials. So I'll run through quite a few snapshots from our work, mostly with fungi, but I'll be mentioning bacteria as well. Fungi as geoactive agents, and you can call this geomycology, fungi as geoactive agents, a subset of geomicrobiology. run through bioweathering, bioleaching, and biomaterials. Some examples using silicates, zinc silicate and sulfide, lead and uranium biomineralization, aspects of phosphate solubilization, biofertilizers and revegetation, and then concrete biodeterioration and its relevance, nuclear decommissioning, and then finally some cultural heritage and an example of the Lewis chessmen that you might be interested in, and then finally some aspects of sulfate-reducing bacteria that we used to work on several years ago for many years and had quite good success in metal bioprecipitation and industrial applications of that process. Okay, so I'll start off with fungi. Now, the main significance of fungi is in aerobic environments. The vast majority are highly aerobic, so soil, rock, mineral surfaces, plant root zone, etc. Just like bacteria, they're found ubiquitously. They're the most dominant organism in soil, for example, the largest biomass, but also in extreme conditions. There's Norman's Law, and there's some lichens. Lichens are a fungal growth form. They're pioneer colonizers of barren rock and surfaces in the early stages of soil formation. You can find them, sand dunes, the filamentous hyphae may be holding the sand dunes together. Toxic conditions, they can withstand a whole lot of different environmental extremes. So you kind of get them everywhere. They're ideally suited because this filamentous growth form enables them to explore the environment, to find nice places to live. They can signal from different parts. They have similar sensory mechanisms as we do, which is why fungi particularly used as prime experimental models. They release extracellular enzymes and metabolites for nutrient release. They also form very important mutualistic symbioses, mycorrhizas with plants, with plant roots. Over 80% of all plant genera are mycorrhizal. So mostly when you see plants, they're not just plants, they're really plant fungal symbioses. And in fact, if you take the bacteria into account, they're really plant microbial symbiosis. There are also biomechanical components of substrate penetration when it comes to rocks and minerals. They can penetrate cracks and grain boundaries and things like this, and just as tree roots, and you've seen this coming up through tarmac, they can also do this on the microscale. And in fact, some of the highest pressures inside living organisms can be found in some fungal systems. Now, if you're thinking about fungi in the environment, you're usually considering them in terms of some kind of decomposition, leaves and organic matter, wood, etc. But, there's other elements involved besides carbon. Majority of all stable elements can be associated with biomass, as essential elements, and also ones taken up accidentally as pollutants and things like this. So if you see something like this, you might think straight away it's some kind of carbon cycle or a nitrogen cycle. You've got decomposition of material. Some of the nutrients and elements will enter the soil pool, some be released to be taken back up again by the plants and other biota. Some will be lost in the groundwater, etc. And some, rain and physical things are affecting things. But this could also be a cycle of anything. This could be a calcium cycle, it could be a potassium cycle, or it could be a radiocesium cycle. Anything that's in that biomass will be part of this cycle. So you can see even just something as apparently mundane as decomposition of organic matter really leads to cycling of all component elements on this kind of local but therefore global scales. Now, as well as the organic environment, they're in the inorganic environment, if I can put it like this, rocks and minerals, and you're very familiar, limestone and sandstone, etc. If you look at the outside of rocks, they're frequently weathered. Things grow, there's plants and all kinds of things. Ultimately, of course, the fate of rock is to become soil. And you can see here the back from this limestone cliff, the gradual subsumation of the limestone into the soil. If you look at the outer crust, you'll see a jungle of organisms. You'll see phototrophs, cyanobacteria, microalgae driving the system, fixing carbon through photosynthesis, actinomycetes, bacteria, fungi, etc. There's some fungi boring and tunneling. And you'll also see this kind of thing, mineral production or secondary minerals where they might be dissolving, in this case limestone and redissolving, reprecipitation, new secondary minerals or reprecipitation. All these kind of things are going on. So, next time you see a rock, whether it be on a building or in the environment, if you think of it as a bit more than a rock, always in biology at school, a rock is an example of a non-living thing, for biology. Well, there's plenty of things on that rock. And so what are the main things that the organisms are doing? Well, it's actually quite simple. It's really a balance between mineral dissolution and formation, or solubilization and immobilization. If a fungus is contacting rocks or minerals, they're excreting metabolites, various substances that can release things from those rocks and minerals, such as phosphate, if it's a phosphate-containing mineral. They can release the metals from that rock or mineral that can form complexes. These can join in the environmental pool of metal ions and complexes, but also react with other components in the environment, which can reprecipitate and form minerals. They can also act as nucleation sites for the metals, which can, by purely chemical and physical chemical means, nucleate and form fine-grained mineral materials deposited on the biomass, and also a whole variety of mechanisms for intracellular uptake of metals anyway, because a lot of metals are essential, such as copper and zinc and magnesium, manganese, etc. Now you can see the balance of dissolution. There's some red lead oxide. There's one fungal colony there. You can see the zone of dissolution where it's clearing this lead oxide, and the same for cuprite, which is on a black background, so you can see this zone of clearing. The organisms are producing organic acids, in this case, just dissolving those substances, and they're quite tolerant. With mineral formation, in other circumstances, they can reprecipitate the dissolved metals, and some examples of minerals that can be produced are oxalates, carbonates, phosphates, and oxides, and you'll see some of that in the next few slides. So firstly, if we consider bioweathering, well, weathering is the breakdown of rock and minerals into smaller particles, eventually leading to mineral soil formation. And bioweathering just refers to the role of organisms in that process. Obviously, there's sort of physical and chemical, rain and thunder and wind and all the rest of it. But bioweathering, the activities of microbes and other organisms, biocorrosion for metals, it's a very important process because it's the primary source of essential elements for living organisms. So the elements have to be released from all that is locked up in the rocks, plants taken up, eaten by herbivores, etc., to enter the cycle of life. So it's a very important process. It also affects decay of rock and mineral-based structures, so therefore buildings, cultural heritage, as we'll see. Some work, we've done this with silicates, which are the largest group of minerals, about 90% of the minerals on the Earth's crust, and most research is not concentrated on any biotic, because apart from diatoms, which have a silicaceous shell, it's not really in most biological systems. However, it can be very important because silicates, amongst other things, give rise to clay minerals, which you will have heard of throughout soil. And there was little, at least demonstration of a fungal role in this, but you can quite see clearly here some simple experiments with muscovite, white mica, that you can get. There's very, very thin layers. It's a layered mineral. You can split it apart. It gives very flat, thin layers, so you can use it like a microscope cover slip and expose it to organisms or bury it and things like this. And quite easily you can see fungal footprints where organisms have etched the surface, they've dissolved. There's some physical things perhaps as they grow along, roughing up the surface, a zone perhaps where there's exudates and things dissolving. And this kind of system, quite easy to show effects of the organisms on minerals. And of course, in that case, quite easily, confirm a role for fungi in formation of clay minerals. Another kind of silicate that Jinjin and Janwei have been working on is the zinc silicate and sulfide ores, because these are used for mining, and this is some work we've had in collaboration with Thailand. This is a big zinc mine, big zinc smelter, Padang zinc smelter Tak in northern Thailand, so the biggest in Southeast Asia. And that's the zinc silicate, actually. It's like some kind of soil, actually. It doesn't look like the usual impression of an ore. And in the silicate, you can see there's a variety of zinc ores, and there's the zinc sulfide, which is the other thing that might be used for zinc. It's got sphalerite. Zinc sulfide is the main ore. And we're interested, because there's a lot of problems there, as we'll see, some leaching and changes. Fungi can change these recalcitrant materials and solubilize them, but reprecipitate as some insoluble minerals, zinc oxalate, calcium oxalate, and also calcite from the calcium that is also present in those minerals. What they can also do is bioleach the zinc sulfide. And This is called chemoorganotrophic leaching to reflect the metabolism, because organic acids are the main mechanism. But why this is important is that this is going on in the environment as well, and a lot of these other things, all these elements can be associated with these kind of ores. So if you get this in the environment, you can get some kind of acid mine drainage or leakage or something like this, even from landfill sites, that might contain lead or cadmium or arsenic, for example. In growth, the fungi can even take up the particles from solution, and you can see them here. They've completely absorbed the particles from solution. But they'll also release zinc, in this case, into solution, and this just reflects the zinc going up to about 20 mg per litre, about 0.3 millimolar, so that's quite a high concentration. And while you might not be interested in recovering zinc so much, this is a good example of why there's a lot of interest in this at the moment. Currently, there's big interest in the European Union, and well, in fact, everywhere, about the critical elements that we have to power the digital age, the so-called e-tech elements. And you can see here, there's the strategic e-tech elements, this sort of bubble here, 14 critical raw materials identified by the European Union, because they're either running out, the supplies are running out elsewhere, or there's geopolitical issues where they may not be running out, but about 90% of these are held in 3 countries, South Africa, Brazil, and China. So of course you've got to keep friends with these countries, otherwise you might have your supplies restricted or the price will go up, etc. So there's a greater need for recycling and protection of existing resources. And you can see in those regions there's some elements, and then you can see up here other metals as well, great economic importance. NERC, Natural Environment Research Council, has just got a program. We have 2 catalyst grants under this. We're just preparing bigger proposals under this scheme, where you can see on the periodic table the various e-tech elements and valuable elements are illustrated. And these are the ones where scientific opportunities are highlighted, particularly rare earths and cobalt, tellurium, selenium. And bioprocessing is always one of the options that is favoured. There's another perspective on knowing about metals and recovery, is the growth of megacities, the migration of populations, and all the problems that causes with use of resources, recycling, etc. And there's the concept of urban geochemistry, studying the geochemistry of metals in the urban environment, the impact of dusts and things like this, everywhere containing metals, on human health, and now a concept of urban mining, recovery of metals from urban waste, batteries, computers, phones, for example. In a ton of mobile phones, there's more gold than in a ton of gold ore, for example. But nobody ever sort of returns or collects the phones. You just keep them in your drawer at home. Recycling, and always, again, bioprocessing is, again, seen as an option to consider with this. The other aspect of this is sometimes you can recover the metals, and when you recover them, they might be in a useful form. And many years ago, you can see 1991, in a project to try and demonstrate recovery of silver, because there was a worry then, photography, ancient times, Stone Age, using kind of film and silver and all the rest of it, to silver from the effluents. It seems, well, it was another century now. We found that fungi, as well as bacteria, can easily reduce silver to elemental silver. So this is an electron micrograph, and all these little dots. It's actually nanoscale silver. But unfortunately, it didn't have the intelligence to call it nanosilver or something then, because that's what it was, a completely different perspective. And you all know now that nanosilver is in all kinds of products, chopping boards and socks and things like this, because of its antimicrobial properties. These are some elemental selenium and sulfur nanoparticles that we produced on some sulfate-reducing bacterial biofilms many years ago, and as part of a project to look at metalloid oxyanions remediation. And there's some from Chan Wei Li in the lab now, some nanoscale cadmium carbonate precipitated with fungal metabolites. And of course, so nanotechnology or nanobiotechnology, you can even say when there's living organisms involved, small particles, reactive, all kinds of purposes and potential. So next are some aspects of lead transformations. Now lead, you all know about the toxicity of it. This is in southwest Scotland at Dumfries and Galloway, lead hills. Always, even disused lead mines, there's always spoil, vegetation, this kind of thing that doesn't grow. And Jung Yoon Dri has been working on this, as well as effects on metallic lead. Even on metallic lead sheeting, you can see biofilms, evidence of things growing, and you can see this little tracery here, where snails or something like that have been going along eating the biofilm. You can even imagine a little biotic lead cycle going in there where any lead might be transferred to living organisms. There's some X-ray imaging with lead shot, and you can see even around the lead shot, there's evidence of some changes in the lead shot. And if you look closely, again, some interesting minerals forming. You can see there's some fungal hyphae on the lead shot. They're quite tolerant of it. They can grow all kinds of minerals, some approaching nanoscale. So we're quite interested in what these were. We thought they were just simple corrosion products, but using X-ray powder diffraction and looking at the elemental composition, there's an X-ray map showing the dominance of lead, chlorine, and phosphorus and oxygen in these minerals, and we eventually identified it as paramorphite, which is a kind of lead phosphate. And interestingly, this is the most stable lead mineral that exists in the Earth's surface. And in fact, it's the desired final solution that's often proposed by, say, soil people to remediate lead-contaminated soil. And this was achieved by adding some kind of cheap phosphate to lead-contaminated soil, like apatite, bone meal. Apatite's really a calcium phosphate. But this was the first time, living organisms shown to mediate this paramorphite formation. And in fact, it's quite intriguing that in the mineralogical surveys of the Lead Hill areas, when the geologists looked at all the minerals there, and they recorded paramorphite, in the soil, that could have arisen through biogenic action, because, any kind of organism that can release phosphate and affect minerals and stuff can make paramorphite. And this was quite good, and we got a nice feature in Nature, highlighted in Nature on that, and subsequently Young has shown this to be able to happen in the field and also in liquid media, so it provides a means of removing lead from solution, actually. When you get publications in some of those high-impact journals, you get a lot of publicity on the web and all kinds of places, and most of it is very nice and reflective, and you can see there's some here from all these great places. You also get some very interesting kinds of publicity that sometimes is quite baffling. So this is quite a good one, advertising, lead pollution, fungi might be our friends, and this is the women's t-shirts, environmental friendly gifts. And so, you can apparently get women's t-shirts that tackle lead pollution and all kinds of things like this. But what is quite weird is this must have been retranslated, so you can see it doesn't actually make quite sense. And so I'm supposed to have said, I'm supposed to have pronounced, lead is customarily regarded as a flattering, fast substance. So now I don't know whether the fast, and of course in the articles we could often spoke about lead is used, gets in the environment. One thing is fishing weights and things like this. And I think there's a few crossed wires with this one. Mushrooms could be used to fight weight pollution, and it was next to all kinds of things about weight loss and slimming and etc. So, and then this was quite an interesting missive from somebody in Portugal who has been suffering from various complaints. Actually, most of them looks like what we all suffer from every day. Actually, sort of feeling unwell, blocked nose, hard of hearing, and things like this. Started in 1976 when I was accidentally shot. And so I'd be very grateful if there's any possibility of help. And then this one, from American, please put something on YouTube explaining this research. I cannot seem to get it across to the range managers here in USA, particularly the ones at the NRA. I think they do not want to hear it. Who would have thought the NRA would be so not subject to taking notice of anything anybody says? But anyway, seriously, on firing ranges everywhere, actually, Switzerland is particularly notorious. Colossal amounts of lead. You would not believe. It's like six-figure tons going into the environment constantly. So a new phosphate mineral. Funnily enough, we did find the same kind of thing before in another big study that we had with depleted uranium, which again is very interesting. There was little known about its geomicrobiology in the environment. And again, we found that fungi could mediate the formation of fantastic new minerals. You can see here various coronoid minerals. This one, Bovaria caledonica, like coral, and these holes are where the hyphae have nucleated, and died, and there's the fantastic minerals. These are new biogenic minerals of the uramphite group, chernochovite, uramphite, They're well known to, nuclear scientists and geologists, this kind of thing, but not known to be able to be biogenically mediated before. But again, here are the organisms doing that, mediating new minerals. And funnily enough, these minerals are, again, actually more stable than the depleted uranium they were attacking. So those were phosphates again. Now, we can consider phosphates separately as well, because if you've got a mineral phosphate, you can consider the metals, but also the phosphate release. And when minerals are dissolved, if that substance is a nutrient, then that is very useful for plants and other organisms. And this is the case with phosphate, exactly the same mechanisms. And Charlie's been working on this. Some inorganic phosphates, there you can see some tricalcium phosphate. Organisms can dissolve, release the phosphate. They can also penetrate and tunnel, affect it. And then you can see some minerals as well. They can make oxalates, calcium oxalate in that case. And there's some of her results where she's been trying to use some fungal inoculum to assist release of phosphate from inorganic sources like rock phosphate to barley. Barley is a low mycorrhizal responder, so it really relies on its own efforts, or the rhizosphere organisms, to get its nutrients. And there you can see with rock phosphate preparations, growth better, and here with the tricalcium phosphate, growth of the barley was much better, and more phosphorus was taken up by the shoots. Well, there's a lot of interest in biofertilizers, particularly in developing countries that might seek ways to more cheaply enhance crop growth. And there's some work to show you that we had from in Pakistan and Vietnam, a British Council scheme is sponsored, and there's some solubilizing organisms, they're bacteria in that case. And we've had all kinds of trials and experiments in both countries, with sugarcane and maize and these kind of things. And you can see, some of the places are very, very good to do those trials on a large scale. And there are some of the workers making, most watching, the one person digging, preparing the compost to actually prepare in Vietnam. This is the one preparation which they can give to farmers that contains a whole lot of different things, including nitrifying bacteria and all this kind of thing. And there's some of the maize fields, one of the trials, and they had some very good results. It was about 30% increase in yield, actually using the traditional fertilizer, but with the biofertilizer. And there's some of the leaflets that were produced in Pakistan to give to farmers to kind of educate them into roles of bugs and biofertilizer and basically these things and how you could affect the plants. And there's some of the team in Pakistan. And going back to Thailand, so this relates not to biofertilizer but revegetation. There's this zinc smelter again, and it looks very, very pretty. I mean, this is actually full of toxic metals. There's a settling pond. The mud is full of metals. But still, there's plenty of life, plenty of plants. And we were quite interested in this Lindenbergia that grows everywhere, even right down to the water's edge. And so this was one project that we had with some of the team, growing in, away in the lab and in greenhouse and in the field, all these kind of experiments. And one thing that we found, again, the mycorrhizal situation seems to be very important. Mycorrhizal inoculation really seemed to affect survival. And you can see there some of the stained roots. You can see the avascular mycorrhiza, ramifying through the root cells. And what we found was that there seemed to be this hard crust of, biomineralized around the roots that you can see here under the SEM, and there it is under a kind of light microscope, where really a lot of zinc was bound and other metals as oxalates, carbonate, phosphate, was bound materials, seemingly perhaps making some kind of protective barrier, to the plant to stop translocation to the plant shoot. They're very interested over there, and in fact a related project is they're trying also to revegetate deforested land, and sometimes this land is caused by something as soundingly innocuous as pineapple farmers. Apparently the pineapples are very bad because they grow the pineapples, but when the rains come, it washes all the soil away, and then eventually you have no soil and probably no pineapples. But if you try to get things back to, what was this kind of like? And there's one of the classes that we were lecturing to in this Royal Development Centre in Phetchaburi province. You can see a very attentive audience. And one thing that they do as well is grow this grass, Vegetar grass that's got very powerful roots, and it can penetrate this hard baked soil. It's almost like concrete. Split it up, then allow bugs and things to get in and do their tricks. And there's one of the slides to educate. And these are people who they employ to work and look after these parks and things like this. And funnily enough, a lot of the people there are police. They're the police force. They put the police force in charge because they think it's important. But of course, the police force don't know much about forests and soil and things like this, they really are policemen. So they're trying to train up, some further knowledge of how to look after the plants and the soil. So there it is in Thai. A teaspoonful of soil can contain more microbes than there are people on the earth. Coming back to more temperate climates, and again the mycorrhizas, there's some the usual mushrooms and toadstools you might see in the woods in Scotland in the autumn. Mycelial systems of ectomycorrhizal russula species. You can lift up and you can see this kind of mycelium adhering together. Different colours might mean different species, and you can see some bedrock below or a boulder. And if you were to look at that again, you would see etching and transformation. There's some, a pine seedling. You can see lots of mineral deposits on the hypha, and there you can see a mineral particle etching and some marks from the fungus there. And this is one of Marina's Scots pines. We had quite a lot of work going on toxic metal minerals in the plant mycorrhizosphere. And you can see here there's the mighty Scots pine with zinc phosphate. And zinc phosphate, again, it forms in zinc polluted soil. It's actually a mineral. It's called hopite. And there you can see some of the things if you home in closer on the mycelium. And you can see the sort of crystals in the mycelium. And as we get higher magnification, you can see the hyphae, you can see the extracellular mucilage, the sheath around that provides a nice hydrated framework for geochemical reactions to occur. There you can see a higher level. These solid crystals containing carboxylate, zinc-bound to carboxylate groups, so possibly oxalate. The fungus, again, altering the geochemical nature of that root environment or mycorrhizosphere. Concrete deterioration. So these effects on rocks and minerals, obviously concrete is a mineral-based substrate, and this is very important for structural materials, but also in the context of nuclear decommissioning and containment and things like this. That's actually a piece of the barrier concrete from Chernobyl. It's a Chernobyl piece of concrete, and in fact the sarcophagus there, it's absolutely riddled with fungi growing all over it. And you can see there's some incubated actually for a long time, about a year, cracking, spalling and breakage. You can actually get much quicker things than that under right conditions, i.e. if things are damp. And here are some mortar samples and cement samples. You can see etching, tunneling. There's tunneling there for some hyphae, some secondary minerals appearing. You can see crystallization around hyphae, making these sort of microtubes and all various things. They can survive quite easily. They can even tunnel. We've even got them going down to several hundreds of microns into the concrete, in apparently adverse environments. And again, secondary mineral formation. In this case, oxalates can be formed, and in fact, the mineral formation also, it's expansive. It forms, and it can also break and accelerate this sort of deterioration procedure. And the calcium oxalates, in fact, their mineralogical names are huolite and wedolite. They're actually, widely distributed minerals in nature because you get them in practically every lichen patina. So how does this bear with nuclear decommissioning? Well, the outer layers of concrete are contaminated in various places in the nuclear industry, and it is just the outer layers that are contaminated. But in decommissioning, you might have to dispose of the whole block unless you get rid of this outer layer. And some traditional methods are a kind of concrete shaver that just goes up and shaves off the outer layers of the walls, or a scabbler that again breaks up the outer layers and collect it. And sometimes these are robotic because obviously these situations might be a bit dangerous. But if we use the sort of this as this model shows, our fungi, our bioactive fungi, our geoactive fungi to deteriorate and dissolve, maybe this could have some kind of role in that. So we developed one thing called the matrix by means of a Scottish enterprise grant, where if you've got the concrete and there's the outer radionuclides and the outer surfaces, biomatrix that can contain the geoactive organisms, which you could feed accordingly, with nutrients and oxygenate, and then the geoactive organisms will be putting their excreted metabolites, the organic acids, protons, etc., into and dissolving the radionuclides that will then diffuse and be taken up into the biomatrix, eventually resulting in loading of the biomatrix where they could become biomineralized, as various minerals precipitated out of solution. And then, of course, then you could get rid of the biomatrix, remove it for recovery or containment in a small volume or disposal by other means. And there's some examples, small examples of these mortar cubes. Using cobalt as an example. Cobalt's one of the radionuclides, but cobalt's quite useful to use because, it's colored, and so you can see removal of this leached, of the color from this leached mortar block, showing removal by the biomatrix that was on the top of this, and that was after a couple of weeks. And we found it also effective for multiple metal contamination. These are the metals that are the main radionuclides of interest in that particular study, cobalt, cesium, and strontium. You can see removal quite effectively. And we're undergoing, after some patent developments, we've done this and, trying, looking for, further commercial development in this regard. And this is the kind of place where we were speaking with some companies at Dundee, where there are places like this, old ponds and storage vessels that were originally filled up with kind of water and had radioactive things in them. And of course, they're all deeply contaminated, the surfaces. And in retrospect, apparently, they kind of think that, well, maybe concrete wasn't such a good thing, to use for this purpose. You wouldn't use it these days. But anyway, that's in the past. But you can imagine, it'd be quite a good test site for a biomatrix, nice and regular, and put on the wall, and you could look after it and see what happens. Unfortunately, also what is permissible is that you could just bulldoze soil over it. And again, this is quite permissible under current legislation. So really, you can pay for a JCB for a day, cover it with soil, and problem solved. Anyway, I better not start ranting about the nuclear industry. Going back to concrete, also, it's obviously important, where we live. So this is just one of my several offices during my 30-odd years in Dundee. Looking out the windowsill, you can see many, many colonies, protolichens, cracking, spalling, microcolonies, blackness, all different colors, fungi, everywhere. There's another characteristic place, such as you might see anywhere where the concrete has actually been painted by a nice special paint. There you can see this black microcolonial fungi bursting their way through everywhere you look at. And in fact, these are ones that are found right away across Europe. They're coming up on marble, especially all the marble statues and things like this. When you see little black dots, they're these fungi. There you can see the pebbled ash, which is a great thing for catching everything. And you can see whenever you see staining and this kind of thing, it's organisms growing. And in fact, this has developed so far that there's moss growing here. And there you can see some moss. In fact, this building is really the early stage of soil formation. If you get long enough, this is how soil forms. There's another characteristic site. Now you can see this bit here, where you can see some leakage and damp and colours. Going closer, there's enough for about 6 PhDs in this particular bit. There's iron oxidizers, probably iron oxidizers, there's alkalophiles, there's calcite coming out, all kinds of different colours, where you can see this outer layer, blackness and growth and darkness, damp collecting, with rain and wind and things will be spalling, weakening, all the rest of it. And so buildings, but also cultural heritage. I've mentioned lichens 1 or 2 times. Lichens are a fungal growth form. There's from a burial ground near us. And there you can see 2007, one of the cherubs, quite happy. He's got a bit of growth on him. 2010, you can see some changes. Some of the lichen has grown, but some has come off. It comes off in the rain and the wind. When it comes off, it takes surface layers with it. You can cause removal. And you can see just the other day, really, he's almost obliterated. He's still smiling, but it won't be long before he's not smiling. And in fact, some people think within 50 to 100 years, all of this kind of stuff, and you're talking about frescoes and monuments and statues and outsides of cathedrals, you name it, may be destroyed unless somebody's looking after it. As well as that, there's obviously growth on plasters and things like this, and even paper, organic decomposition, but you also get mineral transformations. Much of the paper contains calcium carbonate, and you'll even get oxalates found in paper when it's decomposing from all the calcium carbonates and strange metal minerals from the metals in the inks that were used. The last bit on the fungal bits is cultural heritage is this Lewis hoard of gaming pieces, or sometimes called the Lewis chessmen. They're found in Ewig Bay at Ardroyle. They're Viking, 13th century. their walrus tusk. And there's 93 pieces, 11 in Edinburgh, 82 in London. 11 were held back by the guy who flogged them to the person in London, and he didn't give them the full lot, so I think that's the story of that. And there's lots of other stories as well, actually. The boy who found them was supposedly murdered by the ghillie, who he stupidly told after he'd found them, and all things like this. But anyway, all wear a glum expression. 4 of the warders are biting the tops of their shields. This is probably something like a modern equivalent of biting your iPad when the Wi-Fi goes down. And apparently the kings are supposed to have this expression that they're plotting, the scene of the next carnage, and the queens who've got their hands like this, they're supposed to be trying to seek a way to find solace to the occasion. People find all kinds of interpretations, and of course one thing that they were very interested was how these marks and get on these statues, there to inform on their history and their burial. And Jim Tate at National Museum of Scotland in Edinburgh, and Flavia from Rome, and works in Institute of Conservation. And, thinking, well, where's the fungi involved? They say, well, probably, they could be. And sure enough, we got some walrus tusk from private collection. We didn't have, you're not allowed to go and shoot them anymore. You have to actually go through quite a rigmarole to get these kind of things. And my job was sawing up the walrus. And when you saw up the walrus tusk, you have that smell. And the smell is exactly the same as the smell you get when your teeth are being drilled by the dentist. And it's quite a funny sensation when you're doing it, actually, if you've got such a love of dentists as I have. And anyway, there's a mineralized crust, and etching, tunneling, boring, and again, Young did some of this work in here. So quite easily showed that, fungi can be responsible and can attack this kind of stuff, and we've got another cover there. This is also something that maybe should worry you. This is a boar's ancient boar tusk, Flavia got hold of, a little piece on agar with Aspergillus niger, the organism that produces citric acid that's in all the Coca-Cola and Iron Brew and everything else, citric acid producer. This is after 2 days. It's completely and utterly dissolved, completely dissolved. So this shows you what a fantastic substance the enamel is on your teeth. It must be incredibly tough to withstand, because if you didn't have this, at least plenty of young people wouldn't have their teeth left. So maybe that's already happening. So look after your teeth is the moral of that one. Now the last little bit I'll finish off is to do with bacteria, and a particular group of sulfate-producing bacteria that use sulfate like we do oxygen. They breathe using sulfate and they produce sulfide. So you can see here he's letting off his sulfide. And they're strictly anaerobic. So, the fungi aerobic and SRB anaerobic. So you can see that some things can, pass aerobic and anaerobic environments. So they can reduce sulfate to sulfide, and they can grow on organic compounds or hydrogen. Strict anaerobes, and there they are there, some motile little flagellum there. And they're very good for bioremediation, as you'll see, because this relates to the precipitation of metals as metal sulfides. And you get this in wetlands and bioreactors. And there you can see some in kind of stab cultures. The growth is reflected by the blackness, and the blackness reflects precipitation of iron and copper sulfides in that case. Now you can see a TM, the Nagma did this some years ago. Even there's some particles around in the periplasm of the cells. They also produce some bicarbonate, and which keeps the environment quite neutral. And most toxic metals are insoluble, so they come out of solution as highly insoluble sulfides. And this is everywhere. So when you see, estuaries and muds and blackness, and if you can dig down, this is actually by Tayport, the Tay estuary, some of the black muds, you can smell the sulfur smell. This is where they are, SRB, everywhere. And if it wasn't for SRB, locking up all the metals that end up into the oceans and rivers, we probably wouldn't be here. So this is where a lot of metals end up, as insoluble sulfides and sediments in the oceans. So very important organisms. But if you take that process out, obviously it's a way of removing metals from solution as the sulfides. Now one thing that we were interested in was removing from contaminated soil, and in fact we used another group of organisms to solubilize the metals first. Another group of organisms important in the sulfur cycle, which was sulfide and sulfur oxidizing bacteria in a process called chemolithotrophic leaching, which is different to the fungal leaching that I mentioned before that relies on organic materials. Basically, these organisms make sulfuric acid, and you can imagine the leaching effect of that. It basically just dissolves things and leaches out the metals. So if you've got a metal sulfide there, you basically get a metal sulfate, or the metal and the sulfate in this sort of very acidic leachate. And acidic thiobacillus is the name of the main organism, and there's already a very established metal bioleaching industry that's gone on actually since the 1950s, particularly in places like USA, Canada, and South Africa, and Australia, where it's used to recover metals from low-grade ores. About 20% of copper in the USA, for example, comes from bioleaching of low-grade copper ores. And there's one such leaching mine in the USA, producing sulfuric acid. You know, the ore is crushed, etc. Basically, it's watered. You know, it gets acidified, but the organisms naturally occurring, they just grow and they get to work, make their sulfuric acid, leach the metal out, and which is recovered by some kind of electrowinning or scrap iron or something like this. Anyway, we did a similar kind of thing, except the organisms in soil. So we had the sulfur oxidizers in the contaminated soil, where they do the same thing, make a leachate, and then pass the leachate into the sulfate-reducing bacterial system, where the sulfate is reduced to sulfide, and the sulfide will precipitate the metals as solid metal sulfides, thus removing the metal from solution, leaving a metal-free effluent, and something that might resemble clean soil afterwards, or at least a clean product. And apologies, old-fashioned data, I tried not to, but anyway, you can see there, you can see the pH fall as the leachate comes and the sulfate stabilizes. There's an example of leaching from an actual contaminated soil that we obtained that contained those kind of metals, and you can see, cobalt and nickel were actually in this soil. You can see quite efficient leaching efficiency of these organisms. Cobalt, 74%, and copper 69, manganese and nickel 68, for example. And these are just the kinetic of for different metals in the soil, where the metal is released. When the maximum is released, then of course it declines, so you get these peaks of release. The black is the metal that passes out through the reactor. We found really the SRB to be pretty much 100% efficient. Nothing got past the sulfur-reducing bacteria. So it was they're a very effective system, as also is the leaching system for moving stuff about. And this was a project where BNFL and others were involved, and we did get up to demonstration plant level. And this is the soil reactor. There were the SRB reactors of this demonstration plant, and there was a whole rig. And we were quite pleased. We were focused on in the biotech, the focus on biochemical engineering in the FRSE publication of this. And funny enough, pointing out in the article saying about remediation of land and bioremediation. It's a big multi-million industry in the USA, but rather less over here. Well, unfortunately, it still is rather less, particularly in the UK, but that's not necessarily in Europe, as you'll see. Anyway, so this was very nice to be thought of as biochemical engineers. Now, in parallel, others, including Shell, but this moved to Holland, and a company called Pax, has developed this kind of technology, and even from the 90s, it's been in operation. So there's this one product they've got here, Sulfate Q. Again, it removes sulfate and converts it to sulfur, and also recovers metal, as I was saying, as metal sulfides. And this is a huge zinc refinery. It's one of the biggest in Europe, if not the biggest in Europe, Narstar zinc refinery. And so you've got the anaerobic bioreactor. Sulfate reducing bacteria, as I mentioned, is fed on either organics or hydrogen. Hydrogen comes in from natural gas, just a splitter, splits it. And then the metal sulfides can be removed. And there's some zinc sulfide, which is a kind of whitish, yellowish, sort of creamy color. And then, in this case, in contrast to our one, it goes to a sulfur reactor for sulfur oxidation, where they get elemental sulfur, and then they can be recycled. So the first reactor reduces sulfate to sulfide, ethanol and H2 as the energy source. Second bioreactor oxidizes the sulfide to elemental sulfur, just with air feed. So you're getting metal sulfides as a product, you're getting sulfur as a product, you're also getting a clean effluent, and you can recycle the sludge actually back, if you need to the smelter. And there is actually the photograph of this plant. There's been several in iteration. This was from the late 90s. That is the reactor that contains the sulfate-reducing bacteria. So you can see it just looks like a big chemical works, actually. And you can see the scale of it from the person there. So there are actually processes, some of these that are in operation, real commercial operation, and for many years, and very successfully. So just to finish off then, I've told you about some aspects that we've done relating to microbial, metal, and mineral transformations. You can see that it's a very multidisciplinary area. You need all the physical and biological and chemical sciences to throw light on it, and, encompassed by things like engineering and mathematics to assist with, exploitation and application and understanding for that matter. The three main areas are, the natural processes, the geomicrobiology of the environment, element and nutrient cycling, bioweathering, soil formation, structure and stability, plant productivity, biofertilizers. On the beneficial impact side, the application side, you've got treatment of metal and radionuclide pollution, bioremediation, bioleaching and metal recovery, biomaterials, including nanoscale biominerals, and on the adverse side, if you like, biodeterioration of rock and mineral-based structures, biocorrosion of metals, destruction of cultural heritage. And of course, all these have an impact on human and environmental health and well-being. Okay, so I'd just like to thank some people. I'd like to thank Professor Sir William Stewart, who's come to see me today in the audience, for first giving me my job in Dundee back in 1978. So this is my 36th year, would you believe, in Dundee. I've gone through many iterations since. These are some recent people in the lab, and many are here tonight. Jeanne Way is not, but she, I think, takes a piece of Scotland with her because she married a Scotsman from St Andrews as well while she was here. So Jeanne did some work on manganese and silicates. Xinjin's here, which worked on the sulfides and phosphate precipitation. Young, who's not about to throw his first daughter into the Tay, hopefully, but he's just got another baby as well. Did the work on lead transformations. Jale works for the biofertilizers in collaboration with Tim George at the James Hutton Institute. Marina's not here at the moment. Did much of the work on the concrete and biomineralization. Marina's back in Kiev at the moment, and I was asking her about the troubles in Kiev, and she's quite sanguine, and she said, yes, it is rather distracting. So that was Chan Wei and Andrea here as well. Chan Wei is working on carbonates, and Andrea is also working on lead and vanadium, and Martin has helped us a lot over the years, especially with the electron microscopy. And Odobinus is representing his genus here, so one animal at least must have been harmed at some point during the making of this presentation. Okay, thank you very much.
Right, I think we're ready for some questions. We'll ask our speaker here, and all the questions are permitted. Remember, there are no naive or bad questions. All questions are good, and we'll ask people to identify themselves, and there will be a microphone because we would like everybody to be able to hear your questions. So there are 2 microphones rowing here. So could we have some questions? Don't count on me because I'm just a poor physician. I can't do it. Please.
Peter Kendrick. I wonder what work is going on to identify and correlate the deleterious effects on structures and concrete and so on as a result of your investigations. Are people predicting when buildings will become unsafe?
That's a good question. I know there's a woman in Oxford, Heather Viles, in one of her papers, and she's particularly talking about some of the cultural heritage that I might have shown on gravestones and things like this. And she says like within 100 years, everything will have gone. With actual buildings, I don't think really, because it doesn't happen very often, but things fall off buildings, and of course that can be a mixture of physical and chemical and rain. But bio will have some part in it, and I don't think anybody does anything really till things are too late in that respect. I have kind of thought about this. I was actually with another colleague in London last week, and she has some work funded from, I think it's English Heritage or something like this, to do with deterioration of cultural heritage, but this is more to understand, I suppose, some of the fundamental processes. I don't think Scottish Heritage have got enough money. I have thought about that. But no, it's. I don't think anybody's actually doing anything about it. I mean, one thing I do know is that some people try and clean up, as you know, they clean and blast and things, and it might look pretty for a little while, but the general idea is that just makes things worse. You're just spreading the inoculum everywhere. You're making nice fresh targets and increasing porosity, and it won't be long. I mean, some things are actually preserved. You get these patinas of minerals, and you can see it on cathedrals or something like this. Maybe they're all black on one side, and this will be a patina of various minerals laid down. Actually, very protective. So sometimes it might be best to leave that alone, even though it might not look so good.
Okay, there is another question in the third row here on the right, please.
Yes, Professor Salzen, fellow. Are there any natural enemies that will attack or feed on these benign fungi and bacteria?
Yes, well, there are quite a lot of things that feed on fungi. I mean, a lot of insect larvae and things like this, beetle larvae. In fact, some of the best examples would be some of the wood-boring beetles, Dutch elm disease, for example. I mean, they cultivate the fungi in the galleries, and the fungus actually causes the Dutch elm disease, but it's the beetles that spread it from tree to tree. So in fact, there has been some work in the past of even kind of bioaccumulation of metals through the food chain through fungi to insects, and which is quite unusual. Were you thinking of some way you could get some kind of predator to clean them all off a statue or something, to clean it up or something like that? And
well, no, I was just thinking that were there any danger to your use of these in industry? Anything that would interfere with your use of them?
Well, I haven't come across anything in that context. I mean, the problem with a lot of some of the industrial applications is maybe not other things. Is the human consideration. People are still quite suspicious of bio in many contexts. I mean, this is one block that we found that came up all the time with nuclear industry, because obviously the nuclear industry is growing up as chemists and engineers, and they're used to big, tough solutions and things like this. And when you're fed on a relentless diet of Frankenstein and things in the papers about anything to do with anything, I mean, it's changed now. Actually, one of our lead contacts in the nuclear laboratory, the National Nuclear Laboratory, is actually does PhD on fungal oxalic acid production and things like this. So it's all kind of changing. So some kind of thoughts like this were more a barrier to exploitation and development than some of the problems of something destroying it.
I just wondered, because you usually get a balance in nature. You usually get a...
Yes, but one thing, especially if you consider sulfide-reducing bacteria, there's very few things that can compete with that. When you get them there, they're massively dominant. I mean, you can get some other anaerobes going, but they metabolize different things, so they don't even compete. You often get methanogens with them, but they don't compete at all. So they're quite safe. Yeah.
Could I ask you about this e-tech elements? This sounds really interesting. So are we missing an opportunity to recycle and get rich from those?
Well, this idea of getting rich is quite an old one, actually. But I know somebody used to, there was a story that somebody kind of patented the sea, because, I mean, one thing we used to work on is sorption, biosorption. Because almost every biological material, whether it's hair clippings or sawdust or bugs, can remove metals from solution by various sorptive reactions, ion exchange, etc. So somebody tried to patent the sea and basically have some little box of stuff on the beach and just run the sea through it. And of course you'll get out all the gold, all the platinum, everything, because everything's in the sea.
Yes.
And of course it doesn't quite look like that. But yeah, there is a big need. So including the European Union, in the US, you name it, is worried now about this recycling and recovery, particularly because of some of the shortages, but because, especially some of the rare earths, I think some of them The only place is China, for example. So we have to keep friends with China. And, and of course there's the usual thoughts, whether supposing something might happen or supplies are restricted or put on a conservation list. And there is, of course, a big problem in getting things back. So again, I was at one meeting like this speaking in Belgium, and there's a big company there involved, Unicor. And they've got batteries wells set up, because we all know about batteries. Nobody ever throws a battery away again. Now, this place is a place of work. You put them in, all the kids take them to school, they collect and collect and collect. So batteries is fine. But other things, no, like mobile phones and things like this. Yeah. And, so there's a lot of stuff. And then you probably know, like in the dust, just on the side of the road, you can collect all the dust. And, it's full of platinum and palladium, from the cars and all the rest of it. And again, you can use bacteria. They can take it out, reduce, you get the palladium out. There's lots of work on this. And, there are actually lots of things going on. So yeah, at the moment there is. It's quite funny, a lot of things have come full circle, because it used to be, like the silver, it was from a point of view of detoxification, but now it would be about getting the metal, be the main thing.
Okay, any other points, questions, burning questions? Yes, there is one at the back on the right, my right. Yeah, great.
Hi, my name is Gregor McCord. I was just wondering if there's anything form of bioremediation that can be used for plastic.
Used for what? Oh, plastic.
Plastic, yeah.
Well, actually, in a way, plastic's easier, because metals can't be destroyed. All they can be is changed in one chemical state to another. They're always there. So you're either making them soluble to put them somewhere else, or immobilizing them to remove their availability. I mean, plastics can be degraded. I mean, some obviously it might take a long time, but it's not generally the case now. Things can be degraded in a short time if conditions are right. So, plastics will eventually be degraded. And, well, you probably know they put things now into the bags as well as a photolytic attack. So, you can't keep your little Tesco bags for it. They just fall to bits anyway, very rapidly, especially if you leave them outside, So plastics are not so much of a problem in a way if conditions are right. I mean, of course, some things might last. If you sling a plastic bottle away, it might take ages floating around. I mean, but if you bury things, so there is quite a lot of work on this. I mean, some of the problem might be is some of the organisms you need to get, and fungi are particularly good, and some actinomycetes for degrading these, long polymers, because I mean that's what they do. I mean, a lot of them are analogous to the polymers in lignin wood, so they're excreting extracellular enzymes. But sometimes they might need another carbon source, so they need to be fed with something else to, if you like, get the energy to make enzymes as well. So they need to, co-metabolize. But there's many examples all through, especially the Superfund sites all over USA. Polyaromatic hydrocarbons, you name it, can all be cleaned up. Trichlorophenols, anything, by fungi and bacterial communities actually very easily in terms of organics. Some plastics might take a bit longer, but yeah, that's possible.
Okay, I think we've exhausted the immediate question, but please don't go away. I would like to just do a vote of thanks. I think you'll all agree that we've heard today fascinating and truly outstanding lecture. Before I came here today, I naively, as a physician and clinical academic, I thought that bacteria cause bad infectious diseases, and fungi occasionally cause diseases, but mostly are found in the forest and could be either eaten or can poison you. And all this biogeochemistry is completely new to me and absolutely fascinating. So I think we really need to thank our speaker for opening completely new ways of thinking. I must say, from my part of life sciences, this is somewhere at the end that I've never seen before, and absolutely fascinating. I really liked the e-tech elements, as my question suggested. I still think that maybe Scotland can get richer somehow, and we're waiting for you to help us, to lead us there. I think the urban mining, the biomaterials, the lead and weight loss, it was all absolutely fascinating. I think the concrete and biomatrix and decontamination all were great, but the bit I liked the most was when you showed the damp concrete and said there are 6 PhDs in it. I've never managed that all my academic life. So thank you very much for absolutely fascinating lecture, and let's thank you in a usual way. Fabulous. Congratulations. Okay. It will get better.